And now we're coming to our third speaker. It's George. He's a junior lecturer at the Faculty of Medicine and the Institute of Pharmaceutical Sciences at the University of Geneva. He's a pharmacist and holds a PhD in life science from the University of Geneva. During his PhD and as a postdoc, he worked in close collaboration with clinical partners from the Hôpitaux Universitaires de Genève on establishing a streamlined platform that combines patient-derived organoids technology and in silico modeling as a personalized treatment approach for colorectal cancer patients. We are very happy that you are here, George, so the screen is yours. Thank you.
First of all, thank you so much for the organizing committee for inviting me for this talk. So today I'll be talking about new approach methodologies for personalized colorectal cancer treatment. So what are new approach methodologies? These are methods that can be defined as in vitro methods or in silico computational methods to assess the efficacy or toxicity or actually any biological response without relying on traditional animal testing. And therefore, they contribute to the 3R conventions, which are replace, reduce, and refine animal use in the research. So in our lab, we have developed multiple new approach and multiple platforms actually that fall under the umbrella of new approach methodologies. As an example, we have our therapeutically guided multi-drug optimization platform, which we use to optimize synergistic low-dose drug combinations specifically for personalized patients. But also we developed a drug toxicity platform to cross-validate the activity of these drug combinations on non-cancerous models to evaluate the liver, renal, intestinal, or even cardiac toxicity. In addition, we are currently as well working with a computational developed tool, which is the Swiss target prediction, developed by the Swiss Institute of Bioinformatics. We also have in our lab a platform for 3D co-culture, complex co-culture models, where we take 3D cellular constructs, like organoids or spheroids, and we co-culture them with endothelial cells or immune cells, and this to further actually complexify the 3D model. And as example on the video, you can see these are colorectal organoids that are that where we add as well in marked in green splenocytes, and you can see the integration of the immune cells inside of the organoid. And finally, also we have developed a PBPK, or it's physiologically based pharmacokinetic model, to see how these drug combination will act and to predict their pharmacokinetics in the human body. But for the sake of timing, I will focus today on our drug screening platform and in the frame of colorectal cancer. So why colorectal cancer? It's actually the third most commonly diagnosed cancer worldwide, and it's the second cause of deaths, cancer-related deaths in the world. So it starts as a protrusion, an uncontrolled proliferation of cells in the lumen of the colon, and then over time it grows with the accumulation of mutations until it starts to invade nearby lymph nodes or even metastasizing to other organs such as the liver and the lungs. So what are the current treatment modalities for this type of cancer? Is mainly surgical removal, if possible, for the primary tumor if it's localized. And this is mainly from stage 1 to stage 3, but also a combination of targeted therapies along with chemotherapies are mainly actually provided for the patients. However, this approach usually decreases actually the quality of life and does not improve the overall survival of the patients. And for a small portion of patients with CRC, they can benefit from immunotherapy, given some certain biomarkers, if they are present or not. So we can say that generally speaking, a one-size-fits-all treatment approach is used in each of these stages. And the major issue with a standardized protocol is that it fails to identify the cohort of patients that would benefit from a certain treatment or not, um, in order to avoid like serious side effects or even the appearance of resistance. Therefore, the main challenges that we have in CRC treatment are mainly the resistance, low efficacy of a treatment, and serious side effects. So a way to overcome these challenges are synergistic drug combinations. Uh, when we tackle the complex machinery of cancer from different angles using different drugs at very low dose, we will be able to reduce side effects, we will be able to reduce resistance, and if we aim for synergy, we will have an increased efficacy. And our goal will be to optimize these drug combinations in a clinically relevant setting. So this is why in our lab we developed our previously validated phenotypic approach entitled therapeutically guided multi-drug optimization. So it's a method that is not only based on in silico modeling, but it combines both experimental and mathematical analysis. So we, as a very simplified schematic representation, we initiate the screen by selecting an initial drug pool of drugs. These drugs they have different targets upstream and downstream of the cell. And most of these drugs are used in clinics or in late phase clinical trial. For each of these drugs, we start by doing drug response curves to identify and determine the range of activity of each of these drugs. And then we start choosing the input dose that we want to use in the model. And the concentrations that we work with are maximum IC20, which is the concentration of a drug that kills only 20% of the cells. And the second concentration would be half of this. So these different drugs are then tested according to predefined statistical matrices that are called orthogonal array composite design matrices. So if you consider this cubic representation as the full experimental space that you have, all of the different possibilities of the combinations you can test, we only test a very small fraction of it that are given by these points. So these matrices, they look like this. So we have here, for instance, the A drugs. Each line corresponds to a drug that is tested at one of the two doses. And the first half of the matrix actually exposes the linear effects of these drugs on a large experimental space, while the second part, it exposes how the effect of these drugs will vary at different dosage. So we start performing the drug screen simultaneously on cancer and healthy model to guarantee the selectivity of the final combination that we obtain. So after testing these drugs in the lab, we take the experimental data, and then we start modeling it using our computational, our mathematical model, which is based on second-order linear regression. And then we generate a mathematical model that looks like something as you can see on the graph on the right. In very simple words, if you consider in black the activity on the cancer cells and in striped white and green, the therapeutic window, which we define as the difference of activity of these drug combinations or the tested condition on healthy minus cancer. And for our model, we consider a negative regression coefficient as activity or synergy, and a positive regression coefficient as antagonism or loss of activity. So the generated model could give us different case scenarios, like here highlighted in red. These are non-optimal cases for us. It's either high efficacy on cancer cells, but also on healthy cells, or loss of activity on cancer cells while being safe, or finally, no activity on cancer but unhealthy. So for us, our optimal case scenario will be the one highlighted in green, which is activity on cancer and while being safe. So we analyze these data and these models, and then we eliminate non-active or active compounds from our initial drug pool, and then we perform the screen in a 3 sequential rounds of the experiment until the last round. We end up with a 3 or 4 drug combination with an increased efficacy and reduced side effects, as well as reduced resistance, because it contains 4 different drugs that most of the time have different targets. So this has been already performed in 2D cell lines, healthy and cancer, and we were able to obtain actually drug combinations that have been also validated in vivo. But we also took it a step further, and then we performed this TGMO screen on patient-derived material, patient-derived organoids. So what are organoids? I think since this morning we have heard this word multiple times. So as a quick refreshment, these are 3D constructs that are derived from self-organizing stem cell. And these 3D cellular models, they usually keep the same architecture and same characteristics as the tissue they are derived from. It's not a new model. It has been there for now 2 decades at least. But however, we usually do not see how do we get them actually from the first place. So we work in close collaboration with the Hôpitaux Universitaires de Genève and a team of oncologists and surgeons. And us as scientists, we know that on this day, we will receive a call to go and pick up a tumor sample. So what happens is that from the OR, we receive a call that there is a patient and that the specimen is about to fall out. So we run to the OR, and then we take the sample and we bring it to the clinical pathologist. And then at the clinical pathology, they gave us both a piece of the tumor, but also a piece of the healthy tissue that we take back to the lab. And then once we are in the lab, these tissue undergo mechanical actually digestion, but also an optimized enzymatic digestion in order to finally be able to expand, maintain, and use for our drug screen. So in order to be able to use the cellular model for our needs, which consists in a high throughput screen, we needed to optimize a seeding method that will allow us to be able to test up to 155 different combinations. But we needed to obtain a relevant size of these organoids. So what happened is like we were able to optimize a very simple seeding method that allows us to obtain in a 96-well plate a single organoid that varies from a size of 350 to 450 micrometers. And these organoids, each well contains one single organoid. The seeding method is reproducible, and it has been also validated for other tumor types, not only colorectal cancer, for colon cancer. So once we optimized this, we wanted to see how it will go with the treatment. So these are representative images from organoids of 3 different patients that we obtained. One from metastasis, colon metastasis in the liver, and 2 primary colon. And together with our collaborators in clinical pathology and the group of Georg Hosken and EPFL, we were able actually to stratify this patient material according to mutations known in CRC-related genes. And we were able to identify the stage of the tumor, the molecular subtype, and microsatellite stability or instability. These information, it's important to know that this information is not regularly provided by the hospital. When you go and you obtain freshly isolated material, so it takes time, and sometimes it's not even routinely done. And as we can see that here, for these 3 patients, for our study that was published last year, we can clearly see that the cohort of samples, they represent very distinct CRC subgroups. For instance, like the first sample is a CMS4 patient, which is known actually to have a very negative prognosis, while, for instance, the patient, the number 3, is CMS1 with a positive, more positive prognosis. So what we did is that we wanted to treat these 3 patients and see how they will respond to the same treatment. So we tried out one of our previously optimized drug combinations consisting of regorafenib, vemurafenib, palbociclib, and lapatinib. And we treated alongside with the FOLFOXI, which is the chemotherapy standard of care used in the clinics. And the dose that we use here in this experiment is the clinically used dose. And as you can see that for the same treatment given for the patient, each patient actually responds differently, and not only for our combination, but also for the standard of care, which is expected. So because each patient is different, and because each patient requires a personalized treatment, we decided that we are going to expand this platform. And right now, together with my colleague, Dr. Mireia Andreu, from OC, who is also working together in Professor Patrick Meraldi's lab, is that we're expanding this platform to other, to more patients. And the idea would be that we will perform our TGMO screen simultaneously on the patient tumor tissue, but also on his healthy tissue. And the timeline that we are trying to fit in is from 2 to 3 weeks. We will able to send back to the clinics and the oncologists the information about a drug combination that is specifically optimized for the patient. So it takes 5 to 7 days normally from the day of the tumor isolation to generate a patient or patient-derived organoids. And then we perform our drug seeding procedure. We perform our drug screen, the TGMO, if we consider 8 drugs. And it takes approximately 2 weeks afterwards to send the output of this drug screen. So the next step that we are currently actually working on, we are also working in close collaboration right now with clinical pharmacologists, Dr. Jean Terrier and Dr. Yusuf Dali, because the question that we always get is, how do you evaluate actually these pharmacokinetics of these drugs? How can you make sure that it's not toxic, or there will be other interactions in the body, since we are optimizing these drug combinations on ex vivo organoids. So this is why we started this collaboration and this platform of physiologically based pharmacokinetics. So it's as if we are predicting the pharmacokinetic profile of these drugs within a whole organism. So the first step that we work on is to create, actually, for each drug, a compound profile. And in order to do so, we do an advanced literature search for physiochemical properties like the molecular weight, pKa, etc. But also the ADME, the published ADME parameters like the absorption, distribution, and elimination. And once all of these parameters are found, we start creating the compound profile on SimSip, which is one of the softwares that are available for such studies. And then you start simulating the pharmacokinetic profile of this compound in different populations. First, in order to validate the model, we consider a healthy population. And what happens is that we compare our pharmacokinetic profile of one drug with observed points or observed curves. These are data from public and published clinical trials. We can then start analyzing the drug interactions and finally predict the pharmacokinetic profile of a full drug combination. So to wrap up, all of these, everything I said sounds very nice, cool, and easy. However, there's a lot of challenges that are happening behind the scenes. So first is how to respect the clinical timeframes. And by clinical timeframe is with our discussion with different clinicians, and it's the time between the day of the surgery of a patient and the day that the oncologist decides and his treatment scheme. And we are trying to fit in this platform to respect this clinical timeframe. Another point that is also important is the access to the patient material. How often, how frequent would you get these patient material to be able to perform such screen? And we are only working on freshly isolated material. The second point is translation between animal to human. Some of our drug combinations we have previously validated in vivo, so we have pharmacokinetics in vivo. However, to which extent we can translate this in our model to the humans. And finally, the PBVK for a 4-drug combination, this has never been reported before. And because there's no clinical trials with, for instance, 4-drug co-administered together to the patient, this is one of our current main challenge, to compare the profiles that we are generating with literature data from the literature. And finally, it doesn't seem at first not a problem, but primordial condition is a very close collaboration with multidisciplinary actors within a project like this, like surgeon, clinical pathologist, clinical oncologist, mathematician, fundamental scientist, in order to bring them all together and to work in a streamlined way. This represents one important challenge for us. However, for an example, Professor Novak will be giving actually a tandem talk on December 13th with one of our collaborators from HUG, Dr. Jeremy Mayer, to explain to you and to give you an example of such collaboration. So I would like to acknowledge and thank actually my team members from both molecular pharmacology group and from the Department of Cell Physiology and Metabolism, Professor Patrick Meradi, and all of our HUG collaborators. Thank you for your attention.
Thank you very much, George, for your presentation. Very exciting. We have time for one question from the chat.
Yep, thank you. There were a few technical questions which you maybe can ask in the chat, and there was also a general question about such kind of experiments. How do you get back your results to the clinics? Is there a special standardization needed, or is there quality control needed? And do the doctors rely on your data?
So we didn't get them yet to the clinics. However, as I said, we work with close collaborations with all our clinicians. So we have regular meetings with them to show them our findings and to see what do they expect. Is it right? Is it not right? However, we are currently going, hopefully soon, to move forward with this to the clinics. So as I said, like the close collaboration with the clinicians is actually the most important player for a project like this.